The microbiome is a recognized determinant of health. Infants are born essentially with no microbes, and I'll show you evidence for how they acquire an adult-like microbiota. But throughout the main phase of adulthood, the microbiome is stable, as shown in this graphic representation, unless you have something like an intestinal disease like IBS and IBD. And later in life, at the extreme of life, the microbiota becomes unstable again. Infant met and elder met. The aims of this project are, as you can read on this slide, but basically what we were trying to investigate was the effect of birth mode on the microbiota, because the frequency of cesarean section in Mexico is among the highest in the world. The significance of this is that there's increasing epidemiological evidence that children born by C-section are at greater risk for a range of immune dysfunction or autoimmune type diseases, including dermatitis and eczema. The gut microbiota of the infant is shaped by external influences such as diet, feeding mode, antibiotics, prebiotics, and delivery mode. And there are also internal host factors which shape and govern the microbiota. All of the infants in the InfantMet study were breastfed for at least the first 6 weeks of life. In Ireland, the overall rate of breastfeeding is not as high as we'd like, and it tends to stop earlier than we would like. But because we wanted to remove nutrition as one variable, all of the infants had to be breastfed for at least 6 weeks. For example, in this panel corresponding to stool, some of the individuals are dominated by the Bacteroidetes and some are dominated by the Firmicutes. They appear to have a very different microbiota. Strikingly, however, when you look at the same data from metagenomic shotgun sequencing and you ask the question, are the functions different? The answer is essentially not. Because if you can see from the color coding scheme here, even across the comparisons of the exact same individuals, the bacterial functions encoded by these very different compositions are very similar. So you could cynically say, well, okay, the microbiota is different in infants delivered in different ways. Does that have any biological significance? This is a graphic analysis showing that the microbiota is assembling during infancy, is stable over adulthood, and starts to become unstable during old age. Essentially, it was an observational study to determine the composition of the microbiota in 500 old people over age 65. Look at the color scheme, because we had a bunch of people who were living in long stay, which means long-term residential care, and they're color-coded in red. We had an opposite group of subjects who were in the community, living in their own homes, preparing their own meals. And then we had two intermediate groups, people in residential care but for less than 6 weeks, which we call rehab, and then people living at home but going into hospital for minor procedures, which we call day hospital. 90% of the people living in the community are in this lower left-hand quadrant, and 90% of the people in long stay, if you imagine a line drawn through here, are in the top right quadrant. This is another technique I explained yesterday. It's a hierarchical clustering with a heat map superimposed. The people are clustered along, well, they're separated along the top x-axis, and along this axis we have bacteria. So red is the long stay cohort, and this yellow color is, the green color is the community dwelling cohort. So long stay individuals have loads of these bacteria and very little of these. People living in the community have loads of these bacteria and very little of these, based on the red-blue colors. One of the simplest is called Procrustes. It's orthogonal least squares analysis. If there was no association between diet and microbiota, the lines would squiggle all over the page like a kid's diagram. And some of the lines are admittedly quite long. But when you look at the color scheme, you can see that the two variables are separating out quite nicely. So the diet and the microbiota of the community subjects separates them from the diet and the microbiota of the long stay dwelling subjects. their microbiota takes 3 to 6 months to change from a community type to a long-stay type, which is highly suggestive that the diet is driving the microbiota change. And basically you see that people living in the community have a high diversity microbiota, and people living in long stay have a low diversity microbiota. Interestingly, you see the same association with the healthy food diversity index. The healthy food diversity has a decent linear relationship with the 3 phylogenetic indices for microbiota diversity, suggesting that a diverse diet drives a diverse microbiota. As you age, even if you stay healthy, your microbiota changes. And interestingly, that effect has been replicated in recent papers on European cohorts. If you're sick, if you stay in long stay for a long time, your microbiota changes, and these detailed indices show a much more dramatic change from a high diversity community microbiota to a low diversity long stay microbiota. People living in the community who are the healthiest have a community core microbiota and additional bacteria which we've described as beneficial. That's a bit of a license because we don't have direct proof, but it seems that these bacteria are only present in the healthiest people. You can move through a core microbiota into the worst type of microbiota, which is a long-stay microbiota. The greatest difference between a healthy diet and the long-stay-like diet is that the healthy diet has many of the components which you've heard about corresponding to a pre-Hispanic diet. It means lots of unprocessed fruit, vegetables, white meat, fish, complex polysaccharides, relatively little starch. The long-stay-like diet is corresponded by absence of all of those, much higher concentrations of high-fat dairy and higher intake of high-glycemic food and more red meat. Habitual diet correlates with the microbiota, and the microbiota correlates with health. There is a lower gene count in the low-diversity microbiota of long-stay subjects, long-stay patients, doing too much teaching. In our more recent work, we have shown fine detailed microbiota diet clustering in community subjects, which confirms health correlations. Even if you live in the community, if you deliberately eat a bad diet, you're going to have a lower diversity microbiota, and there will be associated deteriorations in your clinical metadata. I skipped the study of omnivores, vegans, and vegetarians, but I can paraphrase it in one sentence, saying that we can see different microbiota in vegans, vegetarians, and omnivores, and we can relate the differences to dietary ingredients and specific microbiota components. And on that note, I will conclude, and thank you for your attention.